Hey guys, Holland Chambers Biology here, bringing to you genetics lecture number 3, which deals with incomplete and codominance. Now, let's take a step back and think about Gregory Mendel, okay? He studied the pea plants and he found dominant recessive traits. He was able to distinguish monohybrids, capital letters being dominant, recessive traits being a lowercase letter, Punnett squares, the whole bit. But using Mendel's knowledge of inheritance, he was not able to figure out through his research the variations and the mixed traits that he would find along the way. So for example, he couldn't explain why some flowers would suddenly turn pink when their parents were, one was white and one was red. Or, for example, if a chicken was born gray. When its parents, one parent was an all-white chicken and the next parent was an all-black chicken, where did gray come from? That's an entirely new color, new trait, and he didn't know what to do with that. So today let's go and talk about how this is actually possible using the Mendelian genetics. And so the first one we're going to talk about is that mixed trait, which is gray or pink, and this is known as incomplete dominance. Now, incomplete dominance, the key here is that one allele, which remember your capital R's or lowercase r's, neither one is completely dominant over the other. So there really is no dominance. They result in what we call a blend. Okay, so we end up having that pink or gray. So here's statistically what's actually happening. So we have red and white are creating this pink. Now some textbooks will show it to you guys like this. So white is dominant, red is dominant, and then pink is a blend of the two, even though neither one is dominant. So they do the capital W's or even lowercase w's, and they go across and they mix the letters. Okay, so just recognize that. The thing though is that in this class, we're not going to be mixing letters. We're going to keep all the letters the same. So if the trait is color, we're going to be using one letter. So big R is going to represent white, little r is going to represent red. It's no longer dominant recessive because neither one is dominant. Okay, neither one is recessive. It's just this is now simply a symbol for those colors, and therefore when you blend those symbols together, aka heterozygous genes, that heterozygous will reveal a blended trait. Okay, so what happens when we have a pink flower and we want to cross it with a red flower? So again, you need to come up with your key first. So white, I'm just going to say, for example, is big R. Red is going to be the little r. And so these have to be homozygous in order for them to create a heterozygous pink. Okay? So use this as your key. Blend your pink, big R, little r, with your red, which according to this key is little r. Remember, drop it down, bring it over, and make sure to write out your genotype and phenotypes. Now what's different here than what we did before is I would like for you guys to actually include the 0 because white is still a phenotype, still right there. We have options of white, red, and pink. So you need to say that in this specific generation, we got 0 white, 2 pink, and then 2 red. Okay, so remember also when you guys are writing out your problems, to put a box or square around the genotype, put a box or square around the phenotype, or you can highlight it. Just make sure you guys can see your answers clearly. All right, so incomplete dominance is a blend. Well, what happens when you have both traits show up at the same time? Oops. There we go. Little intermission here, guys. Sorry about that. Okay, so in this particular case, if both traits are showing up at the same time, then what we're dealing with is what we call codominance. So co in Latin means you both are like co-CEO of a company, you both have the power. Therefore, codominance is that you both are dominant. So this is when two dominant alleles are manifested phenotypically separate. They're not blended. They're phenotypically separate, which means both alleles are dominant. So again, in some books, you will see this, black being big B, white, because he's dominant, is big W, and therefore black and white stripes, like on a zebra, would be big B, big W. But in this class, we're going to keep it to one letter only. So we're going to use B straight across. We're not going to be blending two different letters. Now the reason why is because when we start to learn dihybrids, which is a cross between two different traits, color and texture, that second letter actually represents a different trait, and it's going to get really confusing. So in my class, we tend to just stick with one letter, B, representing a color. So we're just, and again, it doesn't matter. You could do little b for black, or we could do big B for black. This is just simply a symbol now. So let's say, for example, big B's black, little b is a symbol for white, which means that black and white stripes on a zebra would be the heterozygous. Okay, so again, big B, little b. So let's do a quick problem. All right, and again, please make sure you guys are writing this out in your notes, so stop the video at any time to write this all down. Today's homework, you guys are going to be dealing with situational problems such as this. So I would like for you guys to cross two peppered chickens, and I would like to know what the statistical probability of the F1 generation is. So the two peppered chickens would be considered the P generation. What we mean by peppered is one feather is black and one feather is white. So this is a peppered chicken. So because the peppered chickens are showing both traits, they would be the heterozygous. And so create your key. We'll just use this one as an example. And then go ahead and put them in the monohybrid boxes. Okay. So I want two peppered chickens, and again, remember the reason why we split these apart, big B, little b, is that this is known as the law of segregation. So during gamete formation, this chicken is big B, little b, but during gamete formation, it's going to donate either a big B or a little b to its egg. Or if it's a male chicken, it'll donate a big B or a little b to its sperm. So when you put the egg and sperm together, these are now our zygote options. Okay, so again, this is not the actual, this is the expected. So we expect to see, this type of ratio, but the chicken could have 100 kids and they all be recessive little b. That's the actual. Okay, but the expected ratio is what we're actually statistically analyzing, expected. Okay, so the genotype, again, start with the first box, cross them off, big B's 1. Next box, we have big B, little b, we have 2, and then little b, we have 1. So once you're done with the genotype, we can actually cover up this box. You no longer need this box, and use the genotype to then reference what the phenotype will be. So then look at the letters here, big B, go back up to the top, verify what it is, which is black, and then you just simply write black 1, okay? Big B, little b, I have 2. Go back up to your key, double check. That's your peppered or stripes, that's 2. And then little b, double check. That represents white, and therefore he is white. Okay? So again, with genetics, we don't always have dominant recessive. We can now have a blend, which is incomplete dominance. Okay, so pink or gray. Or we can have codominance, which means that both traits manifest at the same, or both phenotypes manifest at the same time. Okay guys, let me know if you have any questions in class. Otherwise, this is Holland Chambers checking out.